am a part of the LIM lab in the Integrative Biology department. The project that I have been working on this summer is how overexpression of protein HFQ causes adverse effects to small RNA and mRNA duplex formation. So I know that's a lot, so let's start from the beginning. To understand how small RNA works, it's important to go back to the basics of the central dogma. DNA encodes all necessary genetic information, and then DNA is then transcribed by polymerases into various types of RNA. Messenger RNA is then translated by ribosomes, which are made up of rRNA, into proteins, which can then be employed by the cell. Small RNAs are regulatory molecules that can bind proteins or mRNA. For my project, I dealt with small RNAs that bind mRNAs to form an siRNA-mRNA duplex and thereby regulate gene expression. HFQ is a protein that facilitates siRNA-mRNA duplex formation. It takes on a conformation of a hexameric ring. As siRNA and target mRNA are transcribed, HFQ binds one molecule of each, bringing the siRNA and mRNA molecules into close proximity and making siRNA-mRNA duplex formation more favorable. HFQ can bind either siRNA or mRNA first. The order does not matter, but what does matter is that HFQ binds one molecule at a time and not both at the same time. The kinetics of HFQ binding also sheds light into a more generalized three-component system that follows a similar mechanism. If you have three free molecules, compounds, atoms, etc., that bind one at a time, it can follow this pathway where A may bind X first and Y still floats freely, or A may bind Y first and X floats freely. Then the AX or AY will bind the last free-floating molecule to form the three-component complex AXY. For my experiment, this is what I considered a successful siRNA-mRNA duplex formation because when the HFQ, siRNA, mRNA come together, HFQ will readily dissociate from the group and then form the duplex, the siRNA, mRNA duplex, and regenerate free HFQ, as seen in the bottom there. It is also important to note that X and Y, or small RNA and mRNA in my case, do not just bind each other in significant quantities. Therefore, by studying this kinetic scheme, we hypothesized that if there is excess HFQ, or A on the right, there will be no free-floating siRNA or mRNA, the free X and Y in the second step. So the final complex will not be formed. In other words, there will be no siRNA-mRNA duplex formation. This graph is a visual representation of what I just talked about. This graph shows the amount of duplex formation in differing levels of HFQ. Our lab has previously shown that in very low levels of HFQ, there is very little duplex formation. This is region A. And as siRNA and mRNA need HFQ to facilitate efficient duplex formation, it is logical to conclude that when HFQ is rare, duplex formation will also be rare. Our lab has also shown that as the amount of HFQ is increased, the percent duplex formation also increases. This is region B. Region C of the graph is the region that I study, where there is excess HFQ. Our model from the kinetic scheme suggests that in excess HFQ, it will be common for each siRNA and mRNA molecule to be individually bound by HFQ. This means that there's no unbound siRNA or mRNA left to complete the duplex formation. This is what I will call the flooding effect of HFQ. The result is that there will be many singly bound HFQ molecules, but very few duplex bound HFQ molecules. In order to test our model, I constructed systems to control the amounts of siRNA, mRNA, and HFQ expressed in the cell. I had two levels of mRNA transcribed, a high and a low, controlled by a strong promoter and a weak promoter respectively. And then I did the same for siRNA. HFQ was under the control of PLAC, which is advantageous for me and my project because we can tune the amount of HFQ that is expressed by adding IPTG. Adding IPTG turns on the promoter and increases the amount of HFQ that is expressed. In addition to this, however, I also had a strong and a weak RBS, which stands for ribosomal binding site. A strong RBS produces a higher range of levels of HFQ. In other words, for the same IPTG concentration, the strong RBS will produce more HFQ than the weaker RBS counterpart. And then GFP is a green fluorescent protein that I used as a reporter for the level of mRNA that is actively expressed. The amount of mRNA that is actively expressed depends on all three molecules, the levels of all three molecules. How much total mRNA is transcribed, how much sRNA is available to interact with the mRNA, and what effect the sRNA has on the mRNA, and how much HFQ is present to facilitate duplex formation, or in my case, hinder duplex formation. And then I measured GFP by fluorescence activated cell sorting, or FACS, a special type of flow cytometry. So RIB is a small RNA that represses the expression of SOD-B mRNA. So this is a repressive system that I studied. When RIB binds SOD-B, expression of SOD-B decreases. Therefore, GFP expression will decrease as sRNA-mRNA duplex formation increases. As HFQ levels increase, the duplex formation also increases, and then GFP expression will decrease. This will occur normally at first, so this is the downward slope. But then the flooding effect in high levels of HFQ, which is in higher IPTG concentrations, will take over, and GFP expression will increase because duplex formation will decrease. The important thing here is that duplex formation and GFP expression are inversely related, and this is why the model for the flooding effect here for the repressive system is an upside-down version of the model graph shown before, because this isn't duplex formation, this is GFP expression. So I tested 4 constructs for the RIB-SOD-B system. They all had low levels of target mRNA, because high levels of mRNA is toxic to the cell. But A and B have low small RNA, and C and D have high small RNA. For all 4 constructs, at high IPTG concentrations, which means high levels of HFQ, the flooding effect is evident by the upward slope. For the higher range of HFQ levels, the GFP expression began to increase at lower IPTG concentrations. This is because the threshold where HFQ is in excess is reached with less IPTG for the higher range HFQ than the lower range. In addition, C and D have higher levels of sRNA expressed, so the range of repression of GFP expression is much larger. To relate this back to our model, all 4 systems started in A, moved to B, and ended up in C as more and more IPTG was added, so as more and more HFQ was present. The high range of HFQ levels showed earlier entrance into C, and we have shown that excess HFQ is causing adverse effects to duplex formation. We also tested an activating system. GLIM-Z is a small RNA that activates the expression of GLIM-US mRNA. When GLIM-Z binds GLIM-US, expression of GLIM-US increases. Therefore, GFP expression will increase. And in this case, GFP expression is directly correlated to the amount of duplex formation. And as HFQ levels increase, we predict an initial increase in GFP levels and then a decrease in GFP levels due to the flooding effect hindering duplex formation. These 4 constructs all have low levels of small RNA. Only B and D, the red and the purple, show significant flooding effect. These both have the higher range of HFQ levels, so they reach the region of excess HFQ. A and C, however, have less HFQ, so they do not. And they are still in the range, A and C are still in the range where it is common to have duplex bound HFQ. These constructs here all have high levels of small RNA. For these, only one construct, B in red, shows the flooding effect. B has low levels of mRNA and high levels of HFQ, and it is the only system that reaches the point of excess HFQ